Let's get right into it. Number 10. Acarus Crossii. In 1837, an English gentleman named Andrew Crosse was carrying out electrical experiments in his private laboratory. Crosse was wealthy and obsessed with electricity. He was not trying to make animals. He was trying to grow crystals by passing a weak electrical current through a mixture of chemicals and porous stone. For the first few weeks, nothing happened, usually a good sign when electricity and chemicals are involved. Then, on the 28th day, something appeared. A tiny white object began sticking out of the stone. Crosse watched it grow. Then it developed legs. Then it moved. Soon, several small creatures resembling mites or fleas were crawling around inside the experiment. This was not part of the plan. Crosse assumed that microscopic insect eggs must have contaminated the stone or liquid, so he cleaned the equipment, sealed the containers, and repeated the experiment. According to him, the creatures appeared again. He claimed they even emerged in poisonous liquids. Crosse did not announce that he had created life. He openly admitted that he had no idea where the creatures came from. The newspapers were less cautious. They reported that electricity had created life from non-living matter. The public responded exactly how you would expect people in the 1800s to respond. They called him a blasphemer. They accused him of playing God. Strangers gathered outside his home to perform exorcisms, damage his property, and shout that he was working with the devil. The creatures became known as Acarus Crossii. Nobody has proved that Crosse created life. Contamination remains the obvious explanation, and his results were never reliably reproduced. But the damage to his reputation was already done. He tried to grow crystals and ended up with insects, angry mobs, and people performing religious ceremonies outside his house. Number 9. The virus used as a treatment. In the 1970s, genetic engineering was still a new and extremely risky idea. Scientists had begun to imagine using viruses as biological delivery vehicles. A virus naturally enters cells and inserts genetic material, so perhaps a modified virus could carry useful genes into a patient and correct an inherited disease. The theory sounded brilliant. 3 German sisters suffered from a rare genetic condition called arginase deficiency. Their bodies could not properly process arginine, an amino acid found in proteins. Without treatment, the condition could cause seizures, developmental problems, and severe brain damage. Doctors proposed an experimental treatment using the Shope papillomavirus, a virus normally associated with rabbits. The hope was that the virus could help the girls produce the missing enzyme. This was an early attempt at human gene therapy, although nobody knew whether it would work. 2 of the sisters received the virus. When a 3rd sister was born, the doctors tried again. The treatment failed. The children did not experience the hoped-for improvement, and one of them reportedly suffered a severe reaction. It showed what happens when a promising idea reaches patients too early. The doctors were not trying to hurt anyone. They were attempting to save children from a devastating disease. But good intentions do not make an experiment safe. Imagine telling a family, the rabbit virus did not cure your daughters, and one of them nearly died from the treatment. Number 8. The flu virus that came back. In 1977, a strain of H1N1 influenza suddenly appeared in the Soviet Union and China. It looked almost identical to an H1N1 strain that had circulated around 1950. This virus appeared to have skipped almost 30 years of evolution. It was like someone disappearing in 1950 and returning in 1977, wearing the same shirt and asking why everyone looked older. The outbreak mainly affected people under the age of 20. Older adults were often protected because they had encountered similar H1N1 strains before the virus disappeared in the 1950s. One theory was that the virus had been stored in a laboratory or used in a vaccine program and had somehow been released. The World Health Organization initially called it natural, but many scientists later argued that the genetic evidence suggested a laboratory origin. Maybe it escaped from a freezer. Maybe it was released during a vaccine trial. Maybe someone made a mistake and then decided silence was the best public relations strategy. Whatever happened, the virus spread around the world. Science does not always need to create a new monster. Sometimes it can accidentally bring an old one back. Before we continue, here is a quick request from me to you. Researching these stories properly and separating documented accidents from myths and internet exaggerations takes a genuinely ridiculous amount of time. And the only way this channel can only keep making videos like this is if the people who watch this far hit the subscribe button. So please take a moment to hit the subscribe button. Not a comment, not a share, just that button, right now. Thank you very much. Okay, back to the monsters. Number 7. The four-winged chicken. Long before modern genetic engineering, scientists were already manipulating animal embryos to see what would happen. One experiment involved an egg with 2 yolks. The membrane separating the developing embryos was removed, allowing them to fuse. The result was described as a small bird with 1 head, 4 wings, and 4 legs. It was a living creature built like someone had copied and pasted half of a chicken. The experiment became part of a field later known as teratology, the study of birth defects and developmental abnormalities. Scientists such as Étienne Geoffroy Saint-Hilaire and his son Isidore deliberately interfered with embryos by changing temperatures, applying pressure, shaking eggs, and altering the conditions in which the animals developed. Their experiments produced creatures with fused bodies, missing limbs, duplicated organs, twisted spines, and extra legs. The work showed that many so-called monsters were not supernatural warnings. They were biological abnormalities caused by disruptions during development. It was also achieved by intentionally creating thousands of malformed animals. Anyone walking into the laboratory without context would have called it a nightmare. The final result looked like poultry designed by a committee. Number 6. The experiments behind Frankenstein. In the late 18th century, scientists became fascinated by the relationship between electricity and life. Italian scientist Luigi Galvani discovered that electrical sparks could make the legs of a dead frog twitch. The frog was not alive, but its muscles still responded to electrical stimulation. To the public, it looked like electricity could wake the dead. Galvani's nephew, Giovanni Aldini, took the demonstrations much further. In 1803, Aldini performed an electrical experiment on the body of an executed criminal named George Forster in London. He attached conductors to the corpse and switched on the current. The dead man's jaw trembled, his face twisted, one eye opened, his arms and legs moved. Electricity was simply forcing the remaining muscles to contract. But that explanation becomes much less comforting when a dead man opens one eye and appears to look directly at the crowd. Experiments like these helped inspire Mary Shelley's Frankenstein. Her fictional scientist Victor Frankenstein assembled a body and used science to give it life. Scientists were learning to make dead tissue move, and nobody knew where it might lead. The warning was Victor abandoning what he created and refusing to take responsibility for it. That idea became the blueprint for almost every science fiction disaster that followed. Create something powerful. Lose control of it. Act surprised when it destroys everything. Number 5. The foot and mouth disease escape. In August 2007, cattle on a farm in Surrey, England, began showing signs of foot and mouth disease. The virus is highly contagious among livestock. It causes fever, painful blisters, and severe difficulty eating or walking. For farmers, outbreaks are devastating because infected animals are often destroyed. Near the affected farms was the Purebright research site, where scientists and a vaccine manufacturer worked with foot and mouth disease virus. Tests showed that the outbreak strain closely matched a virus being used at the facility. Investigators concluded that contaminated material had probably escaped through a damaged drainage system. The virus may then have been carried beyond the site by vehicles, workers, or contaminated mud. The laboratory designed to study and control foot and mouth disease had apparently helped release it. Animals were slaughtered. Farms were placed under restrictions. Agricultural exports were disrupted. The economic losses reached millions. The facility had secure laboratories, trained scientists, safety procedures, and government oversight. The virus had a broken pipe. The pipe won. The danger is not always a dramatic explosion or an evil scientist pressing a button. Sometimes the monster escapes through plumbing. Number 4. SARS escapes from the laboratory. The original SARS outbreak began in 2002 and spread across several countries. By 2003, health officials had largely brought it under control. Then SARS began appearing inside laboratories. Researchers in Singapore and Taiwan became infected while working with the virus. Later, multiple infections were connected to a virology laboratory in Beijing. One infected researcher transmitted the virus to other people, and at least one person died. These incidents did not trigger another pandemic, but they exposed a serious problem. A virus can disappear from the public and still remain alive inside laboratories. It sits in freezers, tubes, and sample containers, waiting for someone to make a mistake. The scientists were not trying to create a biological weapon. They were studying SARS so they could understand it and prevent future outbreaks. There was no dramatic countdown, no flashing red lights, no warning voice saying, "Containment failure in 10 seconds." There were ordinary mistakes, failures in procedure, and contaminated materials. Sometimes they begin with someone skipping one step, because they have done the same task 100 times before. Number 3. Pharaoh's snake. Not every scientific monster is alive. Some merely look alive enough to make everyone step backward. The Pharaoh's snake experiment uses mercury thiocyanate. When the chemical is ignited, it begins producing a twisting mass of ash. Then it grows and grows. Long branches curl across the table, splitting into shapes that resemble tentacles, snakes, limbs, and heads. The reaction looks less like chemistry and more like someone has opened a portal under the beaker. Videos of an extreme version spread online because it looked like a multi-limbed monster crawling from the flames. The reaction releases dangerous mercury compounds, and the residue is unsafe to touch. The original Pharaoh's snake was sold as a firework before people realized mercury fumes were not ideal family entertainment. Safer versions use sugar and baking soda, producing a similar snake that looks less like a demon arriving to collect a debt. The original experiment is a perfect example of science creating something that appears alive, behaves unpredictably, and poisons everyone nearby. It is basically a monster, except the monster is chemistry. Number 2. The Soviet anthrax accident. In April 1979, people living near the Soviet city of Sverdlovsk began developing a mysterious illness. Victims suffered fever, breathing problems, chest pain, and rapid physical collapse. The Soviet government blamed contaminated meat. The victims showed signs of inhalational anthrax, which happens when anthrax spores are breathed into the lungs. People do not normally inhale steaks. A secret military biological facility in Sverdlovsk had been producing anthrax spores. During maintenance, a filter was removed from part of the ventilation system. Then a shift change occurred, and someone restarted the machinery without realizing the filter was missing. A cloud of anthrax spores escaped through the ventilation system and drifted downwind across the city. Dozens died, although the true number may have been higher. The Soviet government denied the laboratory connection for years and continued blaming contaminated food. Only after the collapse of the Soviet Union was the accident publicly acknowledged. What it apparently needed was a large sign saying, "Do not switch this on." The filter is missing. This monster had no claws, teeth, or visible body. It was an invisible cloud released because one safety step was forgotten. Number 1. The Demon Core. In 1945, scientists at Los Alamos were studying a small sphere of plutonium intended for use in an atomic bomb. Instead, researchers used it in criticality experiments. They wanted to measure how close the plutonium could come to sustaining a nuclear chain reaction. The experiments were dangerous, but the scientists often performed them by hand. In August 1945, physicist Harry Daghlian was stacking tungsten carbide blocks around the core. One block slipped from his hand and fell into place. A flash of blue light filled the room. Daghlian immediately knocked the blocks away, but he had already received a massive dose of radiation. He died 25 days later. That should definitely have been the end of the experiments. Less than a year later, physicist Louis Slotin performed a similar test using 2 beryllium hemispheres around the same plutonium core. He held the hemispheres apart with a screwdriver. Scientists called the experiment tickling the dragon's tail, a phrase that should automatically cancel any procedure. The core became supercritical again, producing another blue flash. Slotin pulled the pieces apart with his hands, stopping the reaction and reducing the exposure of everyone else in the room. He died 9 days later from acute radiation sickness. After the second death, Los Alamos finally ended hands-on criticality experiments. This was an excellent safety decision that would have been even better before 2 people died. The demon core did not move, it did not roar, it did not chase anyone. It simply sat on a table, while brilliant scientists repeatedly brought it close to a nuclear reaction using blocks, metal shells, and handheld tools. The real monster was believing nothing could possibly go wrong. So there you have it. 10 times scientific curiosity, ambition, desperation, or ordinary human error created something monstrous. Some experiments produced malformed animals. Some released dangerous diseases. One made a corpse appear to move. Another created a toxic ash creature that looked like it had crawled out of hell. And the most disturbing part is that most of these disasters were not caused by evil scientists. They were caused by people who believed they understood the risks right up until the moment they did not. Which one terrified you most? Crosse's mystery creatures, the returning influenza virus, the invisible anthrax cloud, or the demon core sitting silently on a table while scientists pushed closer to disaster? Tell us in the comments below. And if you enjoyed the video, hit the like button, subscribe, and turn on notifications so you do not miss the next one. Thanks for watching, and I'll see you in the next one.